Okay, so we're going to pick up, we're talking about cytotoxic types of hypersensitivity, or type 2. And we first talked about how an incorrect blood transfusion, right, the wrong blood type being transfused, would lead to one of the examples. And the second example was hemolytic disease of the newborn. Unlike the ABO types of antigens, the Rh antigen is not an antigen that we make an antibody to unless we're exposed to the Rh group. If a woman is Rh negative, she would have the capacity to respond to Rh because it would be a foreign antigen for her. So if she is negative and she has an Rh positive fetus, then the potential exists for her to develop antibodies to the Rh factor. IgG antibodies would be the type that could then cross the placenta and attack the developing fetus. Now, with the first pregnancy, this often does not happen because blood does not usually get mixed between the fetus and the mom until very late in the pregnancy, like when the placenta is pulling away during the birthing process. So during the first pregnancy, the risk of any kind of problem is very low, but upon a second Rh positive pregnancy, the woman that's Rh negative would have antibodies to the Rh that could then cross the placenta and destroy blood cells in the developing fetus. In order to prevent the mom from making antibodies, they would give her RhoGAM. So RhoGAM is really a passive type of protection. So RhoGAM is anti-Rh antibodies, right? And so they give that to mom so that if there are any fetal blood cells in her system, the antibodies would bind them and then they would get cleared before she even has a chance to make antibodies. So that is, RhoGAM is usually administered right before the birth, like during the contraction stage, right before the birth, and right after birth, to make sure that she doesn't develop any antibodies to Rh and thus protect them. Okay, so that's hemolytic disease of the newborn, the second example, and it's also something that comes up during your virtual lab. The third type of hypersensitivity is type 3, and it's immune complex mediated, and this is something that happens as part of the disease process in a lot of autoimmune diseases. So what happens is that antigen that's circulating in your system, and it might be an autoantigen if we're talking about an autoimmune disease, you make antibodies to these antigens and they bind and they form these kind of little network, these little complexes, right, that are fairly large on a molecular level, and they can get lodged in things like your capillary beds, and phagocytes can come and start to clear them up. But the ones that the phagocytes miss can activate complement, and that can do some damage to the tissues inside your capillary beds and inside some of your organs and such. And so they cause systemic reactions. Now, interestingly, glomerulonephritis is a side effect of having group A strep. So this is Strep pyogenes, and we'll talk about this organism later, but it's also known as group A strep because it has an antigen that's on its surface that's called the Lancefield A antigen. If you allow your immune system to clear group A strep on its own, so meaning you're not taking antimicrobials to clear it out, and you make a wide range of antibodies to this organism, one of the side effects that can happen is glomerulonephritis. And if you have taken physiology, you might be able to recognize some of this terminology that's embedded in this word. So the glomerulus and the nephron are part of the kidney. Itis always indicates an inflammation of. So glomerulonephritis is an inflammation inside the kidneys. Specifically, it's happening in the capillary beds and the glomeruli and the nephron, where these immune complexes tend to lodge, and the immune complex will be strep antigen and antibodies, and it lodges in there, and it can cause a bit of a problem for your kidney. If you are young, right, a youngster that is experiencing this, often they will recover without long-term kidney damage. But as you get older, if you experience this, you have a increased risk of having permanent kidney damage after this. Okay, so immune complex mediated, understand this mechanism of what's happening, and it can actually be something that happens because of a specific type of infection, like with group A strep, it can be an effect, or it could be something that is happening in autoimmune diseases such as lupus or rheumatoid arthritis that's leading to the degeneration of the tissues that are there. Pneumonitis, just one last word, right? This is something similar that's happening in the lungs. So if there's antigens or allergens that are being deposited in the lungs, sometimes you get this immune mediated type of inflammation that's happening in the lungs, leading to pneumonitis. Okay, so this is just showing you 2 autoimmune diseases. So rheumatoid arthritis that's destroying the joints is shown up at the top, and we have systemic lupus erythematosus at the bottom with the characteristic rash. Sometimes they call it a butterfly rash. You can see why it sort of has the resemblance of a butterfly there, but it is the immune complexes that are depositing in the capillary beds of these tissues that is leading to this. But in lupus, this is happening throughout the entire system. It's just visible right here on the tissues of the face. The last type of hypersensitivity is type 4 or delayed cell type. This is the only hypersensitivity that is mediated by T cells. And specifically, we're talking about the cytotoxic T cells that are having the effect. The classic example of this is what is occurring in the tuberculin skin test, where they inject a tiny little bit of the antigens from Mycobacterium, the ones that cause tuberculosis, for example, and then your T cells quickly migrate into this area and cause a cytotoxic type of response in the cells that are surrounding this area. So this is a type 4 or delayed type. It's called delayed because unlike type 1, which happens, very, very quickly, like within minutes, this can take longer, right? Like 24 to 48 hours before you see a major type of response. Contact dermatitis is something that you can see with poison ivy, poison oak. So these particular plants have antigens on their surfaces that our T cells tend to recognize and get very overreactive, and you end up with this seeping, weeping, very itchy type of response. Okay, so these are type 4 type of responses. Okay, so now we're going to talk a little bit about graft rejection or organ rejection, and this is also a type 4 cell-mediated type of response. So grafts are transplants, and they can be perceived as foreign. So what's happening here is that the MHC on the graft cells are what's being recognized, regardless of whether there's, potentially antigen being presented or not. The MHC itself is interacting with the T cell receptors because our T cells have not learned that it's not self, right? It is foreign, so therefore the T cells can get very very excited over these MHC molecules and bind to them very strongly and activate the cytotoxic T cells, and then that is going to lead to rejection. All right, so the closer you can make the MHCs match on individuals, the less likely you are going to see this type of reaction. So just to give you some terminology, an autograft is like what might happen if you burn yourself and they have to transplant skin from one area of body to the other, so that's to self. An isograft is genetically identical, so if you were lucky enough to have identical twin and they donated an organ to you because yours was failing, that's an isograft. An allograft, though, is the majority of what is happening in the organ donation system, where you are getting an organ from the same species, but it is not necessarily genetically identical, right? You're getting it from another donor. Even if it is a family member, it's allograft. And then xenografts don't typically work. And this is when you take tissue from one species and move it into the other. And they've tried that. Say, like, take a heart of a chimp, for example, and then transplant it into a dying infant, but it doesn't work. Hasn't worked. The graft is rejected because it's too foreign. They are exploring doing something like humanizing animals, right? So you would grow them up for the specific intent of using the organs to transplant, and those have to be genetically modified so that the animal is expressing human genes as opposed to their own species. Okay, the graft versus host disease is a type of rejection. Also, it's type 4, but this is usually happening when somebody receives a bone marrow. And what happens in this case is that the T cells from the donor, right? So these are grafted, these are donor T cells who have learned to recognize their previous host as self, will find themselves in the recipient, and it's not self, right? So the T cells attack the recipient because they have not learned that that is self because they've been transplanted, and they attack them. So the graft is actually attacking the new host. And typically this has to do with, again, mismatching of MHC. They can only really treat this with immunosuppressive drugs. That also leads to a person that is very immunocompromised then, and they often, if they're immunosuppressed, then they can end up with opportunistic type of infections. So this is a very bad state. They try to improve the success of bone marrow transplants by trying to remove the T cells from any potential donor marrow, but it doesn't always work very well. Okay, so our last video will be on autoimmune diseases and some genetic acquired and genetic types of deficiencies in our immune system. So I'll just cover a few little bits of that on the last video.